Hi, Camilla. Let's bring you up. How are you doing? Hi, everyone. Camilla, I invited you to speak. If you accept, then you will be able to come up here to the stage. Hi, Gilbert. Hi, Brandon. Do you also want to come up? I'm inviting you. Can you guys hear me well? Hi, how are you? Um, oh,
Hi, can you hear me
There
now?
you go.
Hi,
Yeah,
yeah. Can you hear me?
yes, yeah, we
Oh,
can.
perfect.
Thank you.
Oh, sorry.
No worries. Hi,
Hey,
Brandon.
how you doing?
Good. How are you? Meet Dr. Camilla Bellone. She will be presenting today. So yeah, thanks for coming, everyone.
Nice to meet you.
Nice to meet you, Sue, as well. So you tell me, Katarina, when I should start, right?
Yeah. Let's wait a few minutes for let people come in, and I'll ping a few people into the room, and then I'll introduce you, and then we'll start.
Perfect.
Thank you.
I'm inviting some people now. Hang on.
Hello, everybody.
Hi. How are you doing today?
I'm good. I'm good. How are you?
I'm good too. Thank you. Um, it's cold again. Yesterday was like summer in New York, and today it's cold again. It was so weird.
Yeah, it's cold over here too.
Yeah, but yesterday was so I always still think in Celsius. It was like 23 Celsius and really warm. And yeah, today is winter again, kind of winter. Well, so, anyways, hi Monica, you're here too. Thanks for coming. Monica will be also presenting her work in this club, so I'm so glad you come too. If you want to come up and ask questions later on, please come up to the stage. I'll invite you to speak. Hi Shane, hi Iskander, Ingemar, Jette, and Sundar. Welcome. We'll start in around 1 to 2 minutes. So, hi Monica. I'm not sure, maybe you probably don't know Camilla, but meet Camilla. She's our guest speaker for today.
Nice to meet you, Monica.
Oh, she left. I don't know if something happened. Well, hopefully she comes. Oh, there you are. Okay. Okay.
Sorry, I'm still trying to adjust to this platform.
Yeah, I'm so happy you're coming and listening. So thank you.
Yeah.
Yeah, let me introduce you to our guest speakers. And Camilla, people at this time of the day, most people that usually come in the evenings, like from the West Coast and Asia, they'll listen to the replays later.
Okay.
Just to let you know. Yeah, welcome to the Science Society, everyone. We have our guest speaker, Dr. Camilla Bellone. She will talk about her autism research, which is really interesting. And let me give you a little bit of background information. She's an Italian neuroscientist and assistant professor in the Department of Basic Neuroscience at the University of Geneva in Switzerland. And she studied pharmacology at the University of Milano and then found a mentor in Monica De Luca, a neuropharmacologist, where she completed her master's in pharmacy. And then she completed her PhD in the joint lab of Roger Nicole at the University of California, San Francisco. And then shortly after she joined the Nicole lab, Bellone published her first author paper in Neuron, exploring the trafficking of NMDA receptors in hippocampal pyramidal cells in newborn mice. And then in 2007, she returned to Switzerland and pursued her career, and now she has her independent lab. And she received a bunch of awards. Yeah, the 2012 Gertrude von Meissner Foundation Prize and 2014 Foundation du Puy-Pfizer de la Recherche. And sorry, I don't know French. I'm really sorry. So yeah, she is a really wonderful scientist, and I was happy to meet her recently here. And yeah, welcome, Camilla, and thanks for being here.
So thanks, Katarina, for inviting me. It's a great pleasure, first of all, because I discovered the club deck, and it's a beautiful instrument to share science. So today I'm glad to discuss with you about our recent work. And I mean, let's start from the main result we got in this recent paper is that what we demonstrated is that a mutation in a gene that was previously linked to autism spectrum disorder can make the brain more vulnerable to a second hit. And in this case, the second hit was an immune activation. So what I would like to do tonight, with the time that I have, is that to try to drive you through how we got to this result and which were the question we were interested in at the very beginning. So one important aspect that is that autism spectrum disorder is a neurodevelopmental disease that include and is characterized by social communication deficit and repetitive behavior. So there are many genes that has been linked directly to the autism, and many of these genes are coding for protein that are located at the level of the synapses. That is the place where neurons take contact and communicate. So one of the problematic in this research domain is two questions that I was asking. So one was which is at the origin of this social deficit, why the social domain is specifically and somehow very much affected in this patient. And second question is why there is such heterogeneity across different people. That means that even people that show the same gene mutation can have a high heterogeneity in their phenotype. So these were the two main questions and are still the two questions that drive my research. And to answer the first question, so what at the origin of the social deficit, we rely on one hypothesis that now dated a few years that states that the social deficit in autism spectrum disorder may be the consequence of alteration in motivation to interact with other individuals. So basically, what this hypothesis states is that people affected by autism spectrum disorder, they do not show proper orientation toward the social stimuli, so conspecific. They do not like the interaction, and they do not want to neither initiate or maintain this type of interaction. So although this idea was conceptualized in the framework of psychological work, we are neuroscientists. I'm a neuroscientist. I'm working on animal models. So we took advantage of animal model and the possibility to study social behavior in animal model to really try to dissect the social motivation hypothesis. So which are the mechanisms that are underlying this deficit in social motivation in autism spectrum disorder? And this is the reason why we started to look into the mesolimbic reward system, that is really the brain structure, the brain circuitry that is responsible for the reinforcing properties of social interaction. What does it mean? That this system contain neurons that are dopaminergic neuron in the area that is called ventral tegmental area. And when social interaction, when a conspecific arrive, these neurons are activated and release a dopamine in the ventral striatum. So the hypothesis that maybe deficit in this reward system may account for sociability deficit and for deficit in social motivation. So this is what we have done. So in our recent paper, what we have done is that we used an approach where we downregulated a gene that has been linked to autism spectrum disorder, the Shank3 gene, in mice after the birth initially. And so when you downregulate this gene specifically in the nucleus accumbens, in this part of the nucleus, the ventral striatum, what we could do is that we could recapitulate sociability deficit. Basically, what we could do in this case were to use a free chamber test where you can test the preference of the mouse to interact with another mouse or with an object. Normal mouse specifically go to explore a social stimuli, while when we downregulated the Shank3, these were accompanied by this time that we're spending with a social stimuli and object was the same. So they do not show any sociability. So when we look at specific activity of this neuron within the nucleus accumbens, what we realized was that this change in sociability was correlated with an increased excitability of D1 neurons in the nucleus accumbens. This is a specific type of medium spiny neuron that are GABAergic neuron in the nucleus accumbens. So the advantage of animal models compared to human is that we not have to stop to correlation, but we can prove causality. So we're using a genetic tool to manipulate the activity. We could, what we show is that if we could rescue excitability of this medium spiny neuron, we could also, sorry, recapitulate and restore behavior. So in this first part of the study, we really prove the causality between increased activation of this neuron in the nucleus accumbens and the change in sociability in these mice. So when we then use an approach to look, okay, so we downregulate a gene, we change excitability, we change the behavior, why this happening, right? Which is the mechanism, which are the molecular mechanisms. And so this is how we did it in this approach. We use RNA sequencing. So we really extracted the neurons that where Shank3 was downregulated, and we make an analysis of the genes that were change their expression when we were downregulating Shank3. So which other gene change their expression when we change expression of this ASD-related gene? And there came our surprise, because what we could see is that, of course, we were also changing the expression of other genes that were linked to the synapses, but we also proved that there were many genes related to immunosystem process and inflammatory response. So there were the first hint that we had to show that there was maybe some link between the Shank3 downregulation, the sociability deficit, and the inflammatory response. So based on this data, what we did is that we tried to investigate if there was a causality between this inflammatory profile and the sociability deficit. And actually, we took at that time the approach of the mouse model where the Shank3 is downregulated in heterozygous. So it means that only one copy of the gene is altered. And why this was interesting, because while in the complete knockout mice, sociability deficit were already been shown, heterozygous mice are much more heterogeneous, and some laboratory were showing deficit, other not, and the results were really has discrepancy. So we took heterozygous mice where at baseline we did not show in our lab any differences in sociability. And what we did it is we made a challenge on immune activation. So we injected the LPS. So this LPS basically what does it is increase the inflammatory profile and provoke an immune activation. And this is postnatally, right, in adolescent, almost adult mice. And there, what we discovered is that when the mice were challenged with this immune activation, we were able to reveal sociability deficit. So the mice were now impaired in their social function. So that was the link to say that you have the brain that is more vulnerable to immune activation if there is a presence of this genetic alteration. And then what we were able to not only to reveal that, but we were also able to prove the molecular mechanism. So what we did it is that we found that there was one specific gene that was very interesting because we're linking the excitability and the inflammation, that is the TRPV4. This gene code for a channel that is responsible for excitability and is normally activated by change in temperature and by inflammation. So we saw that in heterozygous mice, when LPS was injected, not only they were showing sociability deficit, but these were accompanied by an increase in excitability of the neurons in the nucleus accumbens and an increase in the gene expression of TRPV4. So at that point, we have just to link the behavior, the cellular, and the molecular mechanism. And what we were able to prove is that if we inhibit the protein, the TRPV4, with an inhibitor, we were able to rescue the sociability deficit and the excitability, really proving the causality between all these elements. So now I think that if I want to conclude is that whether our finding not necessarily point to a cause of autism in people with Shank3 mutation, but demonstrate rather that such a mutation can make the brain more vulnerable really to this second hit, such an immune activation, and reveal that there is an important probably time period during which the brain may be more susceptible to this immune activation. Indeed, in this late age when we activate this immune process, the alteration was only transient, where we think that if immune activation happen early on, the consequences can be more chronic. So I think that our study really was really able to give for the first time a molecular mechanism underlying this gene environmental interaction, and open now the route for a new avenue to really to try to understand better which are the signaling cascade that are responsible for that. And I'm happy now to take question, but maybe the things that I think could be interesting to discuss is really this the origin of social deficit, and maybe there is a need of stratifying patients based on the origin of social deficit. So you can show that social deficit can be the consequence of motivation deficit, learning deficit, or even of anxiety, right? So this is very needed. It's very important to really understand that, and then to really realize that is a high heterogeneity, and there is not just one autism but multiple autism diseases, and then there are no one solution that can fit all.
Yeah, thank you so much. This is so interesting, and I really love your work that you show, how you showed it. And it's always things that we hypothesize, and you finally were able to show to everyone this vulnerability and these insults that add on. So, do you think there are other stressors that would elicit a similar cascade of reactions? Let's say, like stress, may be maternal stress or maternal separation, or some drugs or exposure to some pollutants. Do you think, or are you planning on checking more stressors, or do you think they will have a similar signaling cascade?
Yeah, this is a very important point. I think that yeah, so it can be that other stressor can have the same outcome, maybe by a different molecular mechanism, but definitely the endpoint can be the same. The system is susceptible to stress, to inflammation, and I think that what make play a very important role is really the timing, right? So as I say, we did all our manipulation in adulthood, and this already was showing that we're able to acutely alter sociability and excitability of the neurons. So now what we are planning to do is to apply this challenge early after the postnatal development, where we know that there are critical period of development for the reward system, and we think that by doing that we may end up with a more chronic deficit in mice.
So, do you think in the future we could screen, basically, maybe pre-screen already, based on the parents, but that we can maybe generate a screening, and then with children that have a higher vulnerability, to have different approaches when, for example, an infection is happening, to prevent basically the onset, or maybe a more severe autism onset? Do you think that, is it going that way?
Yeah, I think that more and more study pointed toward personalized medicine, right? And I think this is exactly the point of these studies, showing that we need to look more carefully individual people, individual patient, in order to really understand why they develop their profile. So we need to profile the patient much better in order to have and design a specific intervention. So now I'm not a clinician, but I mean, the reason also why I was interested in this work is that speaking with clinicians and with many actually parents, they often say that the symptoms of their children got worse when they had an infection, right? And seems like a trivial one-to-one addition, but I thought about that, and I mean, everyone just really underlined this aspect, right? So when the kids, autistic kids, get sick, their symptom get worse and worse. So has to be some interaction between these genes and the inflammation or stressor. When this happen, whether it can happen in the when during the pregnancy or even after birth, right? So our studies show that it's not just during the pregnancy that this happen, but can happen even after the birth.
Yeah, very interesting. But I want to open up for everyone else to get the chance to ask you a question. So please flash your mic and go ahead and ask your question. Yeah, if right now people are maybe still thinking or not asking, I'll keep asking. Oh, Monica, go ahead.
Hi, Camilla. I'm not an expert with understanding neuroscience, but I was wondering with ASD, and especially since you mentioned that the onset can be during pregnancy or even after birth. It's a more general question. Do you know, or are there any evidences of any epigenetics involved with autism in any way?
So, Katarina, can you repeat? Because I did not hear very well the question. Yeah.
So as far as it was a little bit. I think you're far away from the microphone or something, Monica. Maybe.
Am I?
I think.
Yeah.
Yeah. Now you can ask again. No. Yeah. Thank you.
I guess I was just wondering if there are any, it's a general question, if there are any epigenetic factors involved with ASD, or is it something that's not usually associated with neuro disorders?
So the question is that whether there are other genetic factors that can contribute during the pregnancy.
I think
Epigenetic.
Mm-hmm.
Yeah, I mean that is yeah. There are actually, there was also a study showing that there was also epigenetic modification on the Shank3 gene that can happen and can be correlated or causally linked to deficit at the behavioral level. So I think that epigenetic modification may play a very important role, and I think that overall, I mean, a general comment, I think it's really now the time to put all the piece of the puzzle together, right? I mean, it's very while I mean, when I started, people were really thinking about autism as a synaptic gene, as a synaptic disease, and really focusing on the function of the synapses. I think now more and more we know, less we know, and we realize the complexity. And I think that there are so many factors that can contribute, that maybe the field should interrogate which are the right direction, are the direction where we should go.
I
Yeah.
should posso
Oh,
falar nesse fala não.
oh, sorry, she dropped out. Dennis, Dr. Shah, do you have a question?
Hi Katerina, hi Camila. That was very interesting research. I mean, as much as I know about the TRPV4, I mean, recently there was a research about the endothelial dysfunction and the relationship between the TPRV4 and, I mean, the potential target for the clinical treatment of the age-related vascular system disease. I know about this part, and about the autism. There is a, I mean, link between the autism and gut microbiota, and I think that TRPV4, because it's just sensitized under the inflammatory conditions, and there's too many factors can be included. I mean, such as protease, serotonin, and other kind of factors, they can have a relationship. It was very interesting to me that I heard this perspective out of that.
Yeah, it's interesting what you say, because it's true that when we end up to have the list of the genes, actually I was looking, I did not know about the TRPV4. I mean, totally, it was completely new for me. And there were many genes that were either downregulated, upregulated. And the interesting part is that we were looking something that could link the sociability deficit with the hyperexcitability of the neurons. So we were really looking for channel, right? Ion channel. And also because there is this idea that autism spectrum disorder are not just synaptic diseases, but are channelopathy, so disease of the channels. So this is how we end up to look into the TRPV4. And when I was reading about this gene, and I found this through a lot of work that have been done related to vascular system or inflammation, but very little about the central nervous system and implication for excitability at the neuronal level. So I think that probably is I'm not sure whether this is only happening in the nucleus accumbens. I think that there is more overall a dysfunction of this gene also in the periphery, and will be interesting to really link more and more, like you said, right, this gut and brain, and there is more and more link between the periphery and the central nervous system. We need to have a proof of causality between these two aspects.
Yes, also there was a, report about the methylation dependent silencing of the TRPV4 expression, which was associated with the various pathophysiological conditions. So I think that you have a long way to go, Camila. Hopefully you're ready.
Thanks.
Yeah. Dennis or Brandon, do you have a question?
Sure. Hi, Camila. Thanks for joining us. Really interesting discussion and paper. I was wondering what you see in terms of the future of this work. Like, which direction are you looking? Like, what would your next experiment be?
Yeah. Yeah, actually, so I'm a I was a, I'm a physiologist by training, and become more and more behavioralist and in vivo work, and I realized that in this project to go on, I needed help from expert in more cellular biology and neuron cellular biology. So actually, the nice thing is that after the first finding and after the before paper was accepted, I contacted few people in Europe, and we participate to a call that is Eranet. So we got a grant. We are four that got a grant to really continues working on that direction. And in that respect, we have help from people that are expert in microglia and astrocyte, people that are expert in molecular signaling, and people that are expert in brain organoid and EPS cells. So I think that to go toward this direction and really to tackle the molecular mechanism, and which is the origin of this TRPV for increase, and whether it could represent a important pharmacological target. This is where I would need the help of other expertise. For what concern my part, I think that what we are really into is to understand whether if we change the critical period of a development. So if we apply this challenge early on, whether to see whether we have more chronic deficit. And then in my lab is mostly trying to develop a new task also to really understand the social motivation deficit. So now we have develop an instrumental task where mice have to press the lever to interact with other mice, where we can really look and dissect research reward approach versus reinforcement learning and effort. So we really want to go at the origin of this motivation deficit. So why mice are not interested is that they are not interested or they are not learning. So really to dissect this at the behavioral level, and then to try to dissect that at the circuit level, where my expertise is.
I really appreciate that. It sounds like a very interdisciplinary effort. I find that those tend to be the most comprehensive.
Yeah, I think is I mean nowadays I when I started, I thought that I want to work alone in my lab, my group of people, and really drive the research within my lab. And now I realize more and more how much I like to work with other people in collaboration, and how we need that desperately, right? There are so many expertise that are needed for answering questions, that we need to go more and more toward collaborative project, and just come out from our comfort zone.
Yeah, I wanted to ask if you were planning also to look into other brain regions and other cell types that are also involved in like social anxiety or anxiety type in mice. Are you planning to go into different brain regions, and maybe are there any drugs that you can use to basically try out if there will be a future way of modulating this behavior?
Yeah, so more than other brain structure, I mean, because I would really like and love to really disentangle this social motivation hypothesis, and I think that the reward system is playing an important role in this aspect. And I would like to integrate into that in the nucleus accumbens the input, right? So the nucleus accumbens receive input from the hippocampus, from the prefrontal cortex, from the insula, and from the thalamus. So you could imagine how important is the integration of all this information at that level. So you could imagine then the hippocampus and the information about the social memory, the prefrontal cortex maybe more about the value of the interaction, and the insular cortex maybe more about the internal state. So I think that I would still stay in this system. What I would really love to see whether this is specific, and I don't think so, for the Shank3, or it can be recapitulated in other model systems. So we have analyzed before also in this respect a neuroligin 3 model, and that have a lot of similarity with the Shank model, and it's not weird to think, because they two gene code for protein that are both at the synapses. So one thing that I would love to, if I would have a big lab and a lot of money, is that to really try to stratify also animal models, and maybe stratify them based on the behavioral phenotype, and based on more like a metabolic profile, or the channel profile. So that would be like a way to mimic what people are trying to do in a clinic, right? So to really try to stratify animal model, and then from that stratification try to see whether similar approach for rescue behavior can be applied depending on where the animals stand in this map. So this is will be my ideal plan.
Yeah, it's expensive, but I understand. When I did OCD research, I did 3 models, 2 genetic ones, and then I elicited through optogenetic stimulation of specific inputs to the striatum. Are you planning on if you would do that, would you plan to mix it up with one that doesn't have a different genetic, like knockout, knock in, for like stressor, some sort of stressor during development, comparing it to the genetic models?
Yeah, it can be the two way, right? It can be like a environmental versus gene, but here we show that there are interactions between two, or you could even mix that, in the sense that it would be interesting also to take genes that are very far apart, in a way that take synaptic genes versus genes that codify for something different. And then to see whether there are at that point also differences in their behavior and their phenotype, and then apply on top of that different environmental like stress versus immunochallenge, and see where they deviate from the normality.
Yeah, very interesting. Does anyone have questions? If not, I'll keep asking.
I have one question. It's about, I mean, it seems that you're mostly, I mean, focused on input to the nucleus accumbens, right? Rather than output signaling.
Yeah, so it's interesting both, right? I mean, we just like the way. I mean, I wish we could do everything, and I think you are right. I mean, the output signal is very important, right? We know that from the nucleus accumbens, the neurons go back to the VTA or they go out from the midbrain versus the ventral pallidum. So it would be eventually very interesting to put everything together and study the input output function. And I think that there is an important retrocontrol from the nucleus accumbens down to the VTA, and we hypothesize that this regulation is maybe one that is responsible of a malfunctioning of dopamine neurons, because there is this retrocontrol that is lacking. But these are really still hypotheses.
Yeah, are you planning to do like, you know, electrophysiology approach to look like the probability of the inputs of release probability and things like that?
Yeah, so we have two approaches. So one, we look at the in vivo approach. So we perform a physiology on freely moving mice, and there we look at the activity of dopamine neurons and see how dopamine neurons react during social interaction. Try to see whether there is a dopamine alteration at the origin of this social motivation deficit. And then the plan is to look at the nucleus accumbens and, of course, look at maybe input specific synaptic plasticity that may occur during reinforcement learning that may be altered in autism model. The other approach that we have is also to look at the neuronal dynamic, look with the miniscope at the activity of a population of cell within the nucleus accumbens. So these are all the different type of approaches that we are trying to use. And I think that, I mean, my question generally are starting from a behavioral readout. I always start from a behavior and then try to disentangle the where behavioral alteration can be originated from.
Yeah, so it's really great. Kana, are you welcome to the stage? Do you have a question?
Yeah, thank you, Katrina. Actually, just now I came, so I'm listening few of you regarding the autism. So we'll shoot out the question after few people are talking.
Okay, yeah, that's fine. Yeah, if anyone else of people in the audience have a question, please come and join our conversation. If not, it will just Camilla and me having one. So
So,
yeah, go.
yeah, Katrina. So, do we have the proper treatment? Either it may be a chemical mediators or any genetic material changes to recover the patient which found already autism.
So
Can anyone?
the question is that whether we have any treatment.
Yeah.
Mm. I mean, unfortunately, there are no pharmacological treatment, right, for autism spectrum disorder. From what I know, there are. I mean, you can treat and you can ameliorate some symptoms, and I mean, maybe one question can be, do we think we can cure autism, right? What is autism? I mean, maybe in the psychiatry, one question as a researcher we should ask ourselves is whether it makes sense still the diagnostic view of clinician, or whether we can think about diseases, psychiatric diseases, more like within a spectrum where different function are altered. So just to make an example, I mean, social deficits are common to autism patients, but are also present in schizophrenia patients, where also motivation in some patients is highly altered. Social deficits are present in a depressive patient related to anxiety disorder. So the question is that do we really think we will arrive to cure autism, to cure schizophrenia, or we need to think instead to ameliorate the function that are altered in these patients? So this is a question I ask myself.
Yeah, thank you, Camila. Actually, autism is a type of bipolar disorder, right? So, apart from the social motivational treatment or psychological treatment, most of the drugs which is used to treat psychotropic drugs, which causes it leads to more depressive patients. Cannot actually the pharmacotherapy is lead to the inhibit the social interaction of the patient, particular patient. What I felt. That list of drugs which is given to the symptomatic relief, so those drugs make them to diminishes their social activity. What I felt, whether am I correct?
Yeah, I have really no real answer to that. I mean, for sure there are a lot of reason why you may end up an impairment in social function. Of course, that is one of the function is the more sensitive probably to any challenge, right? But I mean, one thing that I think that helped in the framework of my research is the to understand then there are behavioral intervention in autistic kids that ameliorate social function and cognition if applied early on. And what I found very intriguing is that these behavioral intervention are based on reinforcement learning. So in other words, if you reframe in the motivation hypothesis, is that maybe these patients, they do not find the social interaction so rewarding because they have not learned the association between an action and the reward, what you need for the reinforcement learning. And if you ameliorate this type of associative learning, and if you teach them with this intense behavioral intervention to like the social interaction and to increase the reinforcing properties. In this patient, in some of them, you have an outstanding response. So I mean, even if I'm a pharmacologist, I think that where we should arrive is maybe to really better understand the behavior, to have maybe some behavioral intervention, and maybe design then pharmacological approach that could help this behavioral intervention.
Yeah, exactly.
Yeah, thank you so much for your questions. I don't know if somebody has any question, please raise your hand. Also in the audience, I think we'll go another around 10 minutes. So yeah, if you have a question, please ask them now. And yeah, if not, I'll keep having a conversation with Camilla.
Sure.
I did a project where we submitted a grant where we did maternal separation, and also we're planning to do inflammation. And it was very interesting. We saw in behavior mostly only differences in cognition, and then we did RNA-seq. We sent it out of different brain regions, and the biggest difference that we saw was in the prefrontal cortex, and especially in oligodendrocytes. It was just interesting to me, and I wanted to ask if you, by any chance, just looked at other behavior types too. You know, we run way more behaviors than we publish, I guess. So I wanted to ask.
Yeah, so I mean, for that specific project with the Shank3 plus the immune activation, to be honest, we ran just the three chamber and direct interaction for a question of time. But we run a lot of other behavior in a general knockout where we have an impairment. And I mean, we see that we found that is interesting is that we, this may of interest of you, I mean, we follow the longitudinal cohort of mice, right? So what we did it is that we follow the same offspring from the birth up to adulthood, and we were trying to look at motor function, social function, and anxiety. And what it was interesting that in this model, I mean, there are motor impairment, in particular for the motor learning part. So that could be also related to cognition. But what it was interesting is that there were different time course for social deficit versus anxiety deficit. So the social deficit in this mouse model appear around the P20, while the anxiety deficit appear later around P45. So I thought that this was interesting because you have different developmental trajectories of behavioral function. And the reason also why I found it interesting, because in related to this specific mouse model that is a Shank3, the patient that show alteration mutation in this gene, they show symptoms later in life compared to other autism kids with other genetic alteration. So I found that was interesting to find this also in the mouse model. I think that we would need more and more this type of study where we look at these developmental trajectories of behavioral function, and maybe we can learn something more about the neuronal circuit underlying that.
Oh, that's really interesting. Yeah, we have also Shank mice in the lab. But I mostly looked at compulsion. So, but that's really interesting that it's so similar to human. It's a really good model. Actually, a friend of mine, he developed a model. He's in Portugal now. It's João Peca.
Yeah, I
He
know
was in
him.
the cooking lab.
Yeah, I know him. Yeah. So we don't have the same mouse model. So the one we use is come from Yung Jang from he was at the Duke and now he's at MIT. And so the one we use is a deletion of the exon 4 to 22, while the one that was developed, I think, from Pekka was as a different exon deletion. I think that the shank is super complicated because all these different isoforms and sometimes they compensate. So I mean, for us also was we were struggling at the beginning to try to figure out which was the model that we want to work with. I think that like someone say, right, there are all the models are bad, but some of them are useful. So we need at one point to stick with one model and analyze that. But it's also interesting to compare, because that also, since different exons are implicated, they can tell you a little bit more about the signaling mechanism underlying the deficit.
Yeah, it's a lot of work to compare different mouse models. It's real, you know, it's a lot of work and it's very expensive. When we had the three at the same time, our animal facility bill was crazy. So
Yeah, I can imagine. just like it's yeah, then is the question, right, where we want. This is why I'm saying that sometimes you think whether we should not just aim more and more at research in a consortium where you are much more efficient in screening based on hypothesis and test hypothesis with man work that is stronger than for one single lab.
Yeah, yeah, I agree. The amount of mouse models one needs nowadays to do things like we have then the CFOS ones, and then we have a D1 versus D2 mouse model,
Yeah, it's
and
just
then we
crazy.
cross them, and then you need like even
Yeah,
more crazy.
yeah, no, exactly. So where we should stop, right? That is the big question.
Yeah, exactly. Does anyone have more questions? Please go ahead. Yeah, Brandon, go ahead.
Just one question. Of course, I have no background in any of this, that's why I'm just learning. But uh, the idea of curing autism seems like similar to curing cancer because of many different types. So I was just wondering if there is similarities. Is there a way that you can learn from cancer research discoveries on how to uh, pinpoint some kind of um, way to cure, or is that way off?
I don't know, Katarina, whether you have an answer for that.
Yeah, actually, that's exactly what I did. That was one of my more recent. So I was looking more at epigenetic factors, and so my idea was um, the cells in the adult organism that stay plastic are mostly, I mean, but mostly I thought are neurons and cancer cells. So usually nature just utilizes the same mechanisms again. So and in cancer research there was way more money, so they're way more advanced historically with knowing all the different targets and epigenetic mechanisms that are going on. Um, so I started screening for novel cancer drugs that are small compounds, so they would pass the blood-brain barrier, and looked at a lot of different data sets that are out there. If there's something that overlaps in plasticity, that also overlaps, like when cancer is starts modulating basically the shell, the shape of the cell. And then I found a few and applied them. And so this was what I did for OCD, for these 3 different OCD mouse models. And it worked surprisingly well, because what I would have done if I would have had a lot of money is first screen for all the different gene expression mechanisms that are different when the onset, like when they have this compulsive grooming behavior and so on. But I didn't have the money for that, so I screened for the drugs and applied them. And then I found 2 types, one that makes it worse, so makes the compulsion become worse, and it worked pretty well. And then a counter drug, a counter mechanism, and that made the compulsive behavior like downregulated it. And yeah, I used different from the same lab. The SAPAP mouse model is from the same lab that comes from the Shank, and then I used a couple other mouse models, and it seemed to work fine. So it's interesting that you're saying that it comes maybe from a different reasoning, but my argument is that we like, if I would have the money, I would systematically screen for different novel cancer drugs and apply them in different disease mouse models, and yeah, and maybe find a, a novel, pathway and epigenetic factor that is important, and maybe apply those drugs if they are not too toxic for different diseases. That would be basically my dream future research if I have the money one day.
So we'll be more repurposing, right?
Yeah, exactly. It's just, it's so impressive what cancer research comes up with very specific targets and gene expression mechanisms. For example, one of my other hypotheses is that during cancer formation, the DNA folds in specific patterns like T shapes and so on, G4, um, C, IM4, and so on. And there are cancer drugs developed that just target that, nothing else. It's really impressive what they in the last years came up with. And since those mechanisms are interestingly triggered by magnesium and a few specific ion changes, which happens when you basically excite the cell, right? You have these different ions spikes. So yeah, it's another thing I would be very interested in. And first looking if there's the shape during plasticity as another mechanism, and then also then use these drugs to modulate it. It's really impressive what cancer research has and what we don't have.
Yeah, definitely.
And that's just because of the amount of money thrown at it. Would it be possible at all to kind of like um, overlap or is there any type of um, interdisciplinary research programs that cross
Yeah,
research?
that's interesting that you asked. It's just started the last year, so NIH has a few grants now, but it's the other way around to basically use mental health drugs and check if it can change the cancer, can like target cancer and serotonin pathway and things like that. And but it's just a very novel thing, so I'm hoping that there will be more like this. And yeah, it's just, the perception of mental illness, that it's an actual disease, is very new. So I wish us basically, people are crazy. What can you do? Just, that was kind of the mentality, and that reflected a little bit also in how much research money there was, I think. And it's just very recent that we see it as a disease like cancer or something like that. So that's a difference, I think.
Yeah, and it's true that if you think the the um, the the cost for the society of mental disorder is impressive, but is in the long run. I mean, in other words, people that suffer of psychiatric disorder from very early on, they may suffer for that for the entire life. So the cost for the society is a huge, it's enormous. So I think that should change the mentality, right, of the people to invest money in mental health.
And also the chronic disorders that, start the comorbidity. Like if you have depression and you don't treat it, or anxiety, people tend to drink more, live unhealthy, um, eat unhealthy, like don't sleep well. It like then triggers all kinds of other chronic disease that you can probably, a lot of them relate to mental health disorders at some point in life, and then just behaviors that are not very helpful start at some point.
Yeah, I agree with Katrina that the socio-economic properties playing major role in, especially in this era. The people are more stressed. I found people, those having mental illness, they have more stressed, and their lifestyle, the changing of food behaviors, everything. It not only mental health, also i I found in India there are number of cancerous patient also year by year increasing. So and the main thing is socioeconomic status as well as the food habit. Food habit, the most of the foods are made by the fertilizers, pouring chemicals, and they want to producing more yield. So by using this food, they are automatically uh, become a cancer patient. I found many people in villages, those who are working in the field, they also affected by the cancers like lung cancer and um, uh, colon cancers. So why? Because of this, the change of food habits. So we need to think that is also one of the major factor plays in the healthy of the society.
Yeah, thank you. I don't know, Camilla, if you have time, if Girish has one more question.
Yeah, sure. I can answer one more question. It's fine.
Hey Girish, how are you? I hope I said your name right. I'm sorry.
Hey, Katerina, no, you did. And I appreciate the opportunity to come up here and ask my question. Unfortunately, I joined the room late, but I'm relieved to see that the replays are on. So I'll definitely be revisiting this conversation and Dr. Bellone, your paper. One question, and I apologize if you've already addressed this. One question that sort of arose in my mind as I was listening to this and skimming the paper is the role of the microbiome, whether it be gut microbiome or oral microbiome, and how it influences, autism, the development of autism in individuals with genetic predispositions. um, Leaky gut during a critical time period of life has also been fairly well associated with increased lipopolysaccharide levels in the bloodstream and chronic inflammatory responses. And then, I guess, as a part B to that question, the efficacy of something like fecal matter transplants or microbial supplementation with, I know within the supplement realm, we're not too far advanced in terms of effective supplements, but any sort of shifts in the microbiome and how that may influence the progression or even treatment um, within the context of the autism spectrum disorder. I'd be really interested to hear your thoughts, especially, anything you might have seen even in experiments done with germ-free mice or, these notobiotic models in which certain microbes are transferred to a mouse's gut and how that influences the progression of ASD. Yeah, that's my question, and I appreciate your time.
Yeah, so thanks for the question. I mean, the short answer is that we have not looked into the microbiota, and the influence on our findings. There are, I mean, amazing paper from Mauro Costa Mattioli that have shown the direct implication of the microbiota on social behavior and relative to autism spectrum disorder. So this is a short answer. So I don't know, but a comment I have. I mean, I think we it's something that we definitely need to take in consideration, and I think that it's very difficult sometimes also in terms of social behavior to compare data from different labs. And I think that mostly of the reason reason is because of the different germ and virus that are present in our animal facility, right? So that could explain some of the differences that people see across laboratories. So that would definitely point toward an hypothesis that the germ the environment where the animal are living is definitely influence the internal state and then have an affect social behavior. That I think that it needs to be taken in consideration. The problem is that there are so many factors that we need to control, and somehow, if we would control everything, maybe we would be left with less finding that come by serendipity. On the other hand, we will acquire more knowledge about detailed mechanism. Where is I think we need to find an equilibrium. But I think that if we want to go toward the microbiota and see the influence of microbiota to autism and to social behavior in general, we need to have research that is stronger and use state-of-the-art expertise and technology and ask the right question. And the problem that I have, exactly as I say, is that the problem of reproducibility across laboratory, and how can we really prove a causality between these findings.
Yeah, those are excellent experimental design points, and it's such an interesting question too, because the other thing I'm thinking about, just on a behavioral level, the horizontal transfer of microbes is a real thing as well, in which um, when we interact with different individuals, we're exchanging microbes between one another. Everybody's microbiota is so different and so unique. At the same time, there's this interchange taking place, and especially with something like ASD, where the progression of ASD itself involves more, social aversion. I just wonder what sort of feed forward loop that'd be creating in terms of changes in physiology and how the microbiota is shaped. But yeah, all those points are well taken, and thank you so much for your insights.
Thank you for the question.
Yeah, thank you so much, Camilla, for giving this really great and very interesting and informative talk. I really appreciate it. And
Thank you to
yeah,
you and
please
to
come back.
everyone.
Yeah, please come back anytime to
Yeah,
our club here. And
that,
maybe one day you'll come with updates or
definitely.
your research.
Yeah, thanks a lot to everyone for the question, for the discussion. I really enjoy. Have a nice day.
Yeah, thank you. You too. And
Thank you.
just a couple of announcements. We'll have at 8 p.m. EST Dr. Enrico Rinaldi. He will talk about this research, how he makes models with AI, machine learning, and also with quantum computing of black holes. And it's quite exciting research. And then tomorrow we'll have Dr. Schwartz and talking about his new X-ray computational ghost fluorescence mapping. And in the evening we'll have a study about sleep deprivation, what it does to the brain. And we'll have many more rooms this week with the study with LSD in healthy adults and anxiety linked to gut microbial metabolite we'll have on Friday. So yeah, it's an exciting new week with a lot of interesting speakers. And thank you so much, Camilla. And see you everyone again later or tomorrow. Bye.
Bye, bye,
Thank
bye.
you, Dr. Camilla. Appreciate it.
Bye.
Thank you, Katerina.
Happy International Women's Day wishes to all women.